Okay, thank you. Sorry, guys. Okay, so as I said, that guys, we need to look at this because in order to know which ones have been taken out by the bacteria or E. coli, then definitely we need to depend on the genetic markers and also the reporter gene. But as I said that just now, if you do not sure which one is the genetic markers, which one is the reporter genes in your syllabus, we claim on everything under genetic markers. Okay, so as I said, that's about 99%, you won't take out the plasmids, and about 1% they are unaltered plasmids. So what we're going to do, we have to do the screening process, screening process. So the transformations of the recombinant DNA into superhost cells give a colony, so allow us to do a screening that the clones that contain the desired genes. So how we can do the screening? We do the screenings due to the presence of the genetic markers or reporter genes on the plasmid. So I'm going to show you three different types. Okay, three different types of the plasmids. The one that we have learned is a PBR322. Another one is a PUC19 and also PGLO. So what are so special of them? Basically, if you look at this PBR322, they consist of two antibiotic resistant gene, ampicillin resistant gene, ampicillins, and tetracycline resistant gene. Okay, they're both. Okay, both. So how about PUC19? PUC19 reduced from two antibiotic resistant gene to become one antibiotic resistant gene. So instead of tetracycline resistant gene, we replace this gene with the LACZ. LACZ code for beta galactosidase. And LACZ is a reporter gene. It will report to us. Okay, it will report to us whether success or not. So that's why we call reporter gene. Then what about genetic marker? Genetic marker determine the survival. Okay, we're going to look at it later. We're going to look at it later. Ampicillin, tetracycline. Okay, and how about this PGRO? PGRO have a special protein here. Highlight. It's called green fluorescent protein. Okay, so you're going to give us the GFP. GFP is a protein that's going to give us the fluorescent color, fluorescent signal when we shine it with UV. Okay, when it shine with UV, you're going to give us a laser, you're going to give us the fluorescent. So again, this is a reporter gene. It report to us. Okay, reporter gene, it report to us whether the clone is successful or not. Are you clear? And we do have another one here, ampicillin resistant gene, which act as a genetic marker. So in order to differentiate them, you can see that every plasmid or every molecular vector, they must have two. They must have two, at least two genetic markers or one genetic marker and one reporter genes. Are you clear? How we use it later, I'm going to show you. How we're going to use it later, I will show you. But at this moment, understand that we do know that for all molecular vector, they must have origin of replication. Can you see that? Okay, origin of replication is not shown here, but should have origin of replications. Okay, so then we have done. Okay, then we do have a pair. Okay, you must have at least a pair of the genetic markers. Can you see that? A pair. Either we call genetic markers or genetic markers together with the reporter gene. Can you see that? Reporter genes and the genetic marker. Are you clear? Okay, so in this table, genetic marker is the one that determines the survival. Reporter genes basically report to us whether successful or not. Okay, so in this case, genetic markers for the PBR322 is a tetracycline resistant genes, okay, and ampicillin resistant gene, PUC19 ampicillin resistant gene, PGLO ampicillin resistant gene. So reporter genes in PBR322 we don't have, but PUC19 and PGLO we have the LACZ that will form blue colonies later. PGLO will give a green fluorescent colony later. Okay. Now, again, as I say that if you are not sure, which one is a reporter gene and forget which one is genetic marker, don't worry. Everything claim it as genetic markers in our syllabus, A2 syllabus. Okay. So why we need to do the screening? Because only small percentage of host cell may take up the plasmid. Okay, and some host cell may take up the unaltered plasmid. Okay, and also some may take up the plasmid with the backwards ligations. Okay, so that's why we need to do the screening. So this screening method include what? Include the antibiotic sensitivity. So this one involve the ampicillin. Involve the ampicillin resistant gene, tetracycline resistant gene. Insertional inactivation. Now, what does this mean? Just now I showed you guys, right? If we have the gene, tetracycline resistant gene, so it's going to give us a protein. But if you do the insert with the gene of interest here, so we inactivate tetracycline resistant gene already. We inactivate tetracycline resistant gene already. Okay. Then we do have the chromogenic substrate. Chromogenic substrate means that it gives us the color change. Are you clear? It changes the color if and without. Okay. And also fluorescent protein will give us the fluorescent signal when that is the successful uptake. Okay. So antibiotic sensitivity, basically vector carrier functional selective or selection marker such as antibiotic resistant gene. So it means that if they have the antibiotic resistant gene, they will be able to replicate in a plate that contain antibiotic, okay, or medium. Here, insertional inactivation means that if this approach a foreign DNA we insert within a coding gene, then that particular gene product is not available anymore. Okay. So chromogenic substrates. What's mean chromogenic substrates? So it depends on the enzymatic activity. Just now we use the beta galactosidase. So the enzyme will show us a different color. So the most common example here is the blue-white screening. Okay, blue-white screening. Okay, so now we're going to use these three methods now. Okay, antibiotic sensitivity, insertional inactivations, and also the chromogenic substrate to learn about this, how we can screen for the successful transformants. Okay, so now look at this. We're going to start now. Okay, so first thing, what we're going to do. Okay, so we have the gene of interest in a beaker. What are we going to add? We add our insulin cDNA. Number two, what we add? We add the PBR322. Number third, what we're going to add? We add restriction enzyme. Number four, what we're going to add? We're going to add the ligase. Okay. So in this process, what are the outcome? The possible outcome. The first possible outcome, okay, we're going to get the recombinant plasmid. So recombinant plasmid means with insulin gene. Okay, number two, so means with insulin gene, it can be in the reverse or the correct orientation, doesn't matter. Means as long as with insulin gene. Okay. Or I put this way, okay, insulin genes. Number two, you may get the unaltered plasmid. Mean this one without insulin gene at all. Okay, so we let the reaction take place. Okay, we let the reaction take place, and then second step. Now we add in the host cells, E. coli. Number two, we're going to add in the mixture here. Okay, if let's say in this case we use a chemical transformation, we add in the calcium chloride solutions. We do this, what we call the heat shot. Okay, so what's the outcome if we do this? So the bacteria E. coli in this case. So three possible outcomes. First, no uptake of plasmid. Clear not? They don't take it. So this one contribute about 99%. Or They take in the plasmid, but take in unaltered plasmid. Means that without insulin gene. Okay, number 3, they take in the recombinant plasmid. Okay, so I stop about. Okay, you can take a photo if you need. Okay, then what we're going to do next. Okay, so this is the summary. Okay, so first part and second part. Okay, we have done the transform already. Okay, so these two contribute to roughly about 1%. Very, very minor. Okay, so now let us look at this. We compare first. Okay, let's draw out. If this is a bacterial, in the first case, first scenario, means that doesn't take up, this bacterial does not take up any plasmid. So if they don't take up any plasmids, this bacterial will be killed. Killed by who? Killed by ampicillin and tetracycline. Because they don't take any plasmid, so it means that they don't have the tetracycline resistant gene, they don't have ampicillin resistant gene. So in this case, because they don't have ampicillin resistant gene, no plasmid, they don't have tetracycline resistant gene. Understand? This is first. Now, how we do the differential here? Number 2, if they take up the unaltered plasmid, if they take out unaltered plasmid, so means that they won't be killed. Okay, they won't be killed. Okay, because why? Because they have ampicillin resistant gene, so resistant to ampicillin. They have the tetracycline resistant gene, so they won't be killed in this case. Are you clear? Understand? And the third one, the third possible outcomes here, they take in the recombinant plasmid. So I don't care whether it's in the correct or orientations or not correct, doesn't matter at this moment. So when they take up this gene of interest, and this gene of interest actually is spanned in between the tetracycline resistant gene, correct or not? So means that this bacterial will be killed by tetracycline. Okay, killed by tetracycline. Okay, this won't be killed. Okay, not killed. Okay, but this, the third one, will be killed by tetracycline, but won't be killed, not killed by ampicillin. Because the ampicillin, they still have the ampicillin resistant gene. But tetracycline, they don't have already. Why don't have? Because of the insertional inactivation. Because we insert the gene at the tetracycline resistant gene there. So it means that the tetracycline no longer function already. Okay, so based on this, what we're going to do? So first, understand the concept. We're going to prepare a plate. So this plate contains the medium, the agar medium, so that bacteria can survive. But we add in ampicillin. So this is ampicillin plate. Okay, we call it as ampicillin plate. And this plate we label it as a master plate. It's very important we have to label it as a master plate. Okay, so what we're going to do here is we're going to take this. After transformation, we're going to spread this bacterial onto a thin layer. We spread a thin layer onto this plate. Okay, we spread a thin layer of the bacteria, the transformed bacterial. Maybe transform or not transform, doesn't matter. We spread a thin layer. When you spread a thin layer, we're going to incubate for 24 hours. Incubate. The next day, we're going to see, okay, from the top, we're going to see the form colonies. Okay, so this we call as a colony. Why they form colony? So very simple. Why they form colonies? Why? Very simple. Look at this. This is the plate. So we spread the bacteria there. Okay, we dilute and spread. So what will happen here is those, let's say for this one, they can be killed, okay, by ampicillin. Why they can be killed by ampicillin? Because they don't take in any plasmid, no uptake of the plasmid. If they don't uptake any plasmid, means that they don't have ampicillin resistant gene. So ampicillin will kill them. So kill how many? 99% will be killed. So the remaining one, okay, that can still survive in the ampicillin plate means that they already take in the plasmid. So those survive. This one actually colonies because they survive. When they survive, so means that they can reproduce. So from 1 become 2, 2 become 4. In 24 hours, they grow into colonies. All of them, they are genetically identical. But at this moment, can I see? So what will happen here is these all colonies, we only know that what's the conclusion here? They can survive, right? So it means that they take in the plasmid. Means that they must take in the plasmid already. But at this moment, I can't differentiate. Can't differentiate. I can't differentiate what? Whether it's unaltered plasmid or they take in with the recombinant plasmid. At this moment, sorry, we can't differentiate. Because if as long as they take in plasmid means that they have the ampicillin resistant gene. So it means that they won't be killed by the ampicillin. Are you clear? They won't be killed by ampicillin. So if you view it from the side, so this is a medium. So it forms colonies like this. Colonies, 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 colonies. They form colonies like this. Are you clear? They form colonies like this. Okay, but we are not able to differentiate them. So what we're going to do here next, okay, so this is a master plate. Don't throw away. Your master plate, you throw away, then you die. Okay, you have to restart everything. Why I say this? Because what we're going to do here is we prepare another plate. This plate also contains the medium. But this medium actually contains tetracycline. This plate contains tetracycline. What we're going to do here is we use a sterile cloth, something like do a transfer. Okay, a sterile cloth, okay, just to touch. So it means that some of the bacteria are going to stick on the cloth. And then we transfer here. So bacteria now start here already. Bacteria start. Are you clear? Can I see that the bacteria there? So bacteria will grow. Okay, but the next day when you compare, so you incubate for 24 hours. So this plate we call as a replica plate. Master plate don't throw away. Master plate don't throw away. You touch. So it means that they will grow. They will grow. They will grow. But some of them, you can see that missing colony. The next day, what we're going to see here is, okay, let me do a cut and paste here. So the next day, when you view the plate, so here originally 1, 2, 3, 4, 5, 6, 7, 8. So you can see that maybe only 1, 2, 3, 4, 5, 6, 7, 8. So out of 8, so some of them missing. 2 colonies missing. So why these 2 colonies missing? So these 2 colonies missing because they take in recombinant. This colony actually missing because they take in recombinant plasmid. Why? Because they when take recombinant plasmid means that they do not have tetracycline resistant gene. So they will be killed by tetracycline. Now we can identify already. So what we're going to do here is you compare. You compare between these 2 plates. Then I know. I go back to my master plate. I'm going to choose this one. I'm going to choose this one, and I do the culture, a mass culture, because this one will be the one that actually give us the insulin. Are you clear? Okay, they will give us the insulin. So this is in our syllabus. Are you clear? Okay, so I go through again, guys. I go through again. So first of all, I have the medium. Okay, the medium contain what? Contains the ampicillin. So this one can help us to kill 99% of the bacteria, the E. coli, which do not take up any plasmid. Because they don't have any plasmid, means that they don't have tetracycline resistant gene, they don't have the ampicillin resistant gene, so they will be killed. Okay, so after 24 hours, you can see that they form the colonies. So what's the conclusion for these colonies? These colonies form because they take up the plasmid. So this plasmid can be unaltered plasmid or recombinant plasmid. Are you clear? They take up the unaltered plasmid or recombinant plasmid. Why they can survive? Because they contain ampicillin resistant gene. That's why they must take up the plasmid already. If they don't take up plasmid, they won't be able to survive. But at this moment, we can't differentiate, and we still cannot differentiate between the unaltered plasmid or recombinant plasmid. So what we're going to do from this master plate, we gently touch it with a sterile cloth, transfer it to a replica plate. But this replica plate contains tetracycline. Okay, so what we're going to do here is we incubate for 24 hours. Those that can survive, basically means that they take up the unaltered plasmid, because unaltered plasmid will have the tetracycline resistant gene. So what I'm going to do, we compare between the plates, okay, with the replica plates and also the master plates. We compare, and then you can see the missing colony. So the missing colony are those with the, okay, insulin gene. So we're going to do the culture. Okay, we're going to do the culture. But don't forget here, okay, something out of syllabus, because in this case, before we do culture, before this, we need to check also. What we need to check? Because just now we said that even though it's a recombinant plasmid, it can be in the forward or correct orientation. It can be a reverse orientation. So we do need to check those we can give us the correct orientations, give us the insulins, we culture them. Those that cannot, then we stop. We don't allow, and we don't culture them because they don't produce insulin. If they can't produce insulin, no insulin, then we don't culture them. So this thing will happen 50-50. Are you clear? The reverse and forwards, but doesn't matter. As long as we have one, we can always have the mass culture and the mass productions. Okay, so I summarize here a little bit. Okay, so for PBR322, okay, for PBR322, so the genetic marker here is the ampicillin resistant gene, tetracycline resistant gene. So outcome. So first, if they know plasmid taken out, basically they don't have ampicillin resistant gene, they don't have the tetracycline resistant gene, so it would be killed, okay, by ampicillin in ampicillin plate. Okay. So second, if they take in unaltered plasmid, you take in unaltered plasmids, so it means that they have the ampicillin resistant gene, they have tetracycline resistant genes, so they will survive in both ampicillin plate, which is a master plate, and also tetracycline plate, which is a replica plate. So if recombinant plasmid, okay, so ampicillin resistance here, yes, tetracycline, no. Why no? Because due to the insertional. Inactivation. Can I see that? Insertional inactivations. So this one will survive in the ampicillin plate. Okay, a master plate, but will be killed in tetracycline resistant plate, tetracycline plate. Okay, or replica plate. Okay, understand? So this is how we do the differentiations. Okay, so basically here we all use two techniques. What are the two techniques? Insertional inactivations as well as the antibiotic sensitivity. Are you clear? Clear. So again, you do not need to copy down. I'm going to go through it again. Then you can take a photo. Okay, so this is the first part. You can take out the photo first. So 2 outcomes when you do the ligations, and also 3 outcomes when you do the the transformation. Okay, done. Yeah, okay, you can post it in the groups. Okay, next, how we do this, the first part. Okay, and then we come to the last part here, the summary. How we use the insertional inactivations and also the antibiotic sensitivity, okay, to form, okay, or to check on which one is the success transformance. Okay, done. So you guys have done, taken the photo already. So later we can post it in the groups, okay? Or later, I mean, when you view the video clips, also you can have this, okay? So we can try to answer the question here: the antibiotic sensitivity and the insertional and inactivations in PBR 322. Okay, so from here you can see that. Can you see that? Here, the missing colony. Can you see that? The missing colony. So from here, you can compare between the master plate. This is the master plate. This is a replica plate. You can see that missing colony here and another missing colonies. So these 2 colonies basically will be the one that we can check whether they can produce insulin or not. We don't check so many, right? We check these 2, right? Whether they produce what we call this insulin or not. If they produce insulin, then we continue to have the mass culture. Okay, so you can try to answer this. Okay, you can check the answer later. Okay, so now we come to the second one, blue-white screening. Okay, blue-white screening. So what's mean this blue-white screening? Okay, so let us look at this, the blue-white screening. Now, why we use the blue-white screenings rather than the PUC19? PUC19 plasmid actually is a newer version, versions of the plasmid compared to PBR 322. Why we use a PUC19? Because we want to reduce the use of the antibiotic resistant gene. You try to imagine if this plasmid accidentally spread to the pathogenic bacteria, then pathogenic bacteria were going to resistant to both ampicillin and tetracycline cranol. So we want to reduce the risk of the development of this antibiotic resistant property or strength in the bacteria. We want to reduce from 2 antibiotic resistant gene, we reduce to 1. So what we reduce here, we want to change the PBR 322 into PUC19. So what's so special about PUC19? If you look at the plasmid of PUC19, PUC19 also have 1 antibiotic resistant gene called ampicillin resistant gene. Okay, and they have 1 multiple cloning sites. So happen that this multiple cloning site actually onto this gene called LACZ. Okay, then they do have the origins of replications. Okay, so The registration enzyme that we're going to use here also BAMH1. So BAMH1 going to cleave at the LACZ here so that it's going to inactivate the LACZ gene. Are you clear? They're going to inactivate the LACZ genes here. So what is the function of LACZ? LACZ genes actually code for beta galactosidase. It's an enzyme. So what this enzyme can do? This enzyme actually can convert one type of sugar called X-gal. Okay, convert this sugar X-gal. When you have this enzyme going to convert the X-gal into a blue compound. Are you clear? The X-gals will be converted from colorless. It's a substrate here. Can I say that? Okay, to get a blue compound. So means that if from blue compound means that LACZ gene is function, is expressed. Basically means that no insertional inactivation. Get the idea? So no insertional inactivation. But if you try to imagine, if we put in, okay, right here, if insulin gene is inserted into LACZ gene, so means that no beta galactosidase is produced. Why no? Because of the insertional inactivation. Are you clear? Due to these insertional inactivations, that's why no beta galactosidase. So if no beta galactosidase, so X-gal won't be converted into a blue compound. So means that colony will become white colonies. Are you clear? Become white colony. So this one we can check. If with the insulin gene, white colony. If without insulin genes, we have the beta galactosidase, then LACZ gene is expressed, so blue compound. So what we're going to do here, very simple. Like what we said just now, we take this mixture. So we have E. coli after transform. So we prepare a plate. Now this plate contain what? This plate contain a medium. This medium have what? Have ampicillin. Have what? Have X-gal. Okay, so you spread the bacteria, a thin layer of bacteria onto this plate, and you incubate for 24 hours. So the next day, what we're going to see here is they form colonies. But colonies, you can see you have white colonies, you have the blue colonies. So what's mean this? Okay, analyze the result here. If the blue colonies means that they have beta galactosidase. They have beta galactosidase, so that's why they can convert X-gal, okay, the substrate X-gal, okay, into the blue compound. So that's why the colony stain blue. Okay, the colony stain blue. So what is the conclusion here? If it can form, so conclusion means that no insulin gene. So it means that it's a blank or unaltered plasmid. Are you clear? So no insulin genes, unaltered plasmid. Okay. Now how about this? White color colonies. White color colonies means that they don't have beta galactosidase. Why? Because you already insert the insulin gene. Clear? Are you clear? So means that X-gal, because no beta galactosidase, X-gal cannot convert to blue compound. Okay, so this process even easier. Can you see that by using the PUC19? Okay, by using the PUC19, easier. One-step process, you can differentiate it already. Can you see that? We can differentiate already. Okay, so but basically, those that form colonies means that those are survive, right? So means that they must take up the plasmid already, right? So in order to differentiate between unaltered plasmids or recombinant plasmids, so this one, conclusion, they take up the recombinant plasmid. Are you clear? They take out the recombinant plasmid. Are you clear? Okay, so that's why in this process, you will be able to differentiate. You will be able to differentiate between these two colonies by using the different color. So what are the technique we use? So first, with the ampicillins, we killed 99%. 99% will be killed. So this 99% because of what? Antibiotic sensitivity. Can I see that? Second technique here, because we insert insulin, so that's why you can't form the blue compound, right? So the second technique here is insertional inactivation, because we insert it, so inactivate. And why we give a color? So we use the technique here called chromogenic compound. So by doing this, what is the benefit? We reduce, can you see that? By this case, we reduce the number of the antibiotic resistant gene, decrease the risk of the spreading of this gene to the pathogenic bacteria. Are you clear? And also very simple, you do not need to go through a 2 days process. One shot, we can complete and count everything already. Correct not? You can see that. Okay, so you can take the photo for this one. So the principle first, PUC19. Okay, so next, how we can actually do this? So transform, right? Take this E. coli and then we spread it, okay, onto the plate. So the next day you can see two different, okay, colonies. But 99% already killed, so that's why it forms a small colony. So in this case, we can actually identify. So those colonies, white colonies, what we're going to do, we take it out. For example, this one, we check whether it can produce insulin or not. If can, yes, can produce insulin. Got insulin, then we do the mass culture so that we can have the mass productions of the insulin. Okay, so summary here. So for PUC19, so PUC19 is going to have two, okay, in your syllabus we said two genetic markers, but in fact one genetic marker called ampicillin resistant gene, and another one is the LACZ gene, which is the reporter gene. So what is the outcome? So if the bacteria take up no plasmid, almost 99% of this, so they don't have ampicillin resistant, they don't have the LACZ, so they will be killed by ampicillin. So 99% will be killed. If they take out the unaltered plasmid, because unaltered plasmids, there's no insertional inactivation, so they have the ampicillin resistant, they have the LACZ, so they form blue colonies. Okay, so they form blue colonies because they survive and because of the LACZ. Okay, so because they have LACZ gene, so they have the beta galactosidase to convert the X-gal into the this blue compound. So if they take up this recombinant plasmid, so they are ampicillin resistant because of insertional inactivations, then you won't be able to get the LACZ gene anymore. So no beta galactosidase, then it form white colonies. Okay, they form white colonies. So you can also copy out this one. Okay, basically they survive lah. Okay, they survive and they form colonies. Okay, so what are the technique we use here? So this one, because of the insertional inactivations. So why form blue colonies here? Because of the chromogenic. Okay, chromogenic compounds or substrate. And why the kill here? Because of the antibiotic resistant. Okay, they don't have antibiotic resistant genes, so antibiotics sensitivity. So 3 techniques we use. Okay, so you can take a photo. This is a PUC 19. Okay, PUC 19. Okay. So now, how about PGRO? So PGRO, same concept. PGRO, same concept. Okay. If you see PGRO, the last one, similar concept, but we replace the chromogenics. Okay? We replace the chromogenics substrates with the green fluorescent proteins. So you look at the PGRO. So similar PGRO will have this ampicillin resistant gene. They do have this green fluorescent protein, GFP, the gene code for the green fluorescent protein, and they do have the origins of replications as usual. Okay, so we use the registration enzymes to cut at the green fluorescent protein here to insert our insulin gene. Are you clear? We cut it. Okay, so what we're going to do here is, again, we're going to prepare the plate. This plate contains a medium, and even simple, this medium only with the ampicillin. So the next day, you're going to see they form colonies. Okay, but at this moment, all colonies will be white color. They form all white color colonies. Okay, but if you shine it with UV, some colonies now it form green. It shows a green. Okay, it shows us green. So when you shine with UV, those with the green color basically means that they have green fluorescent protein. Why they have green fluorescent protein? Means that these colonies have unaltered plasmid. The next day, are you clear? Why we shine UV? They have the green fluorescent protein. When I have the signal, means that they already give us the green fluorescent protein, so they take up unaltered plasmid. So because the green fluorescent proteins still remain intact. Okay, but how about white color colonies? White color colonies means that they don't have the green fluorescent protein. Why they don't have a green fluorescent proteins? Because they take up the recombinant plasmid. So means that they have the insulin. So this one, we're going to take it out and then we do the checking. And then if correct, then we do the culture. Okay, so similar idea compared to the PUC 19. Are you clear? Once we use the chromogenics, here we use the fluorescent protein. We use the fluorescent proteins. Okay, so 3 techniques also we use, 3 techniques. So why GFP? Because of the green fluorescent protein. So why we don't have the green fluorescent protein? Here because of the insertional inactivations. And here because ampicillin's antibiotic sensitivity. Clear? Okay? Antibiotic sensitivity. So we use 3 techniques, then we'll be able to actually differentiate. Okay, so summary. You can take a photo first, then before I do a summary for PGRO. Okay, so now we do a summary. Now PGRO actually we have a different way to do it. So this is what we normally do. Okay, but sometimes question may ask you different way. So be careful. PGRO, for PGRO, P-glow, okay? So PGRO will have the ampicillin resistant gene, the gene code for green fluorescent protein, and also the outcome. So if they don't take out any plasmid, no plasmid, so means that they don't have ampicillin resistant gene, so means that they'll be killed by ampicillin. Okay, so if take out the unaltered plasmid, so you're going to have both gene, so it appear as a green fluorescent. Okay, green fluorescent colonies when you shine it with UV. Okay, but if the recombinant plasmid, so they have ampicillin resistant genes, but they don't have green fluorescence, so they'll appear as a white colony, even though you use the UV to shine them. Are you clear? Under the UV lights, they still will appear as the white. Okay, so here the techniques: antibiotic sensitivity, green fluorescence, so this one we use a fluorescent protein, and here white no because of the insertional inactivation. Okay, you can take a photo of this PGRO. Okay, so hopefully that's you guys understand the concept of these screenings. Okay, so generally they can be any methods, but the overall concept is still the same. Okay, the overall concept, they can use any kinds of the way, okay, but they won't run away from these 4 different techniques. Okay, so go back to this blue-white screenings. You can go through it with like Z and beta galactosidase. So this is why I say that. You can see that they form the blue and also the white colonies. Can you see that blue and the white colonies? Okay. And we can use the fluorescent proteins. Okay, you can use a fluorescent protein. So the first fluorescent protein discovered is the green fluorescent protein. We take it from this jellyfish. My bet here you need to actually isolate it. Okay, it's a species name. You have the bioluminescent property. So they exhibit the bright green fluorescent. We expose to blue to a UV range. Okay. So if no plasmid uptake, so it won't have the formation of colonies. Why? It's killed by the ampicillins. If we unaltered plasmid, it will give us a green fluorescent proteins. Okay, why? Because unaltered plasmid will have intact GFP, will give it a GFP with the presence of arabinose. Okay, and we shine with UV. These two we won't be able to actually differentiate until we check it. So either in the correct directions or reverse directions. Again, I say that you won't be able to differentiate them by using the UV, but we can always check whether they can produce insulin for us or not. Okay, so no green fluorescent color was shown, no green fluorescent color was shown because insertional inactivation. Insertions inactivates the green fluorescent genes. Okay, so you can see that in this case, you compare both plates are the same plate. Can you see that in terms of structure, same plates? But if you shine with UV, you can see that some colonies appear as the green color because they already have the green fluorescent protein genes there. Okay, so this is the artwork. Okay, nowadays we have more than one kind of color proteins. More than one kind of color. So we have different ranges, so we can actually do a painting. This is a petri dish, and this is a bacteria. We paint the bacteria on this case and let it grow. It become artworks here. Okay, so that's extra information for you. Okay, so with this, I end for today's lesson. So I stop recording first.